Hello and welcome back to Educator.com and welcome back to Biochemistry. So in our last lesson we talked about myoglobin. In this lesson we're going to start talking about hemoglobin. So let's just jump right on in. Okay. Now, whereas myoglobin is involved in the diffusion of the oxygen in muscle tissue. So whereas myoglobin is involved in the diffusion of O2 in muscle tissue, hemoglobin that's the protein which actually transports the oxygen in the blood, is the protein, excuse me, which transports the O2 in the blood. Now, hemoglobin has 4 subunits. So hemoglobin, which is abbreviated HB, it has 4 subunits. So it's not like myoglobin. Myoglobin was just a single subunit protein that has a binding site for 1 molecule of oxygen. It has 4 subunits, and each of these subunits has a heme group with a heme group. So we have, excuse me, 4 sites to bind O2 per protein molecule. Now, the interactions of the subunits, they cause conformational changes that affect hemoglobin's affinity for O2. Okay, so the conformational changes among these subunits, as one O2 binds, there's going to be a conformational change, and that conformational change is going to affect the extent to which another O2 will bind or release depending on the physiological conditions at hand. So you remember, we need to actually bind the O2 tightly. We need to take it to another part of the body, to the tissues, and we need to be able to release it. So we need that flexibility, that great degree of flexibility, in fact, because the oxygen that's coming, the lungs is going to be a particular concentration of oxygen, but that's not the same concentration of oxygen that's in the tissues. So we don't want some molecule that just binds O2 and hangs onto it. It needs to be able to deliver it, and we don't need it to, if we have a molecule that is, that doesn't bind it very well, if it doesn't bind it very well, then it's not going to take up the oxygen that's in the lungs. So we need that high degree of flexibility based on different concentrations of O2. And again, since it's a gas, we're going to be talking about different partial pressures of O2. Okay, so now hemoglobin has alpha and beta subunits. So it has 2 alpha subunits and it has 2 beta subunits. Alpha and beta subunits, 2 of each. So we have an alpha 1, beta 1, and an alpha 2, beta 2. So the alpha 1, beta 1 are, they interact rather tightly, and the alpha 2, beta 2, they interact tightly. Now, there is interaction between the alpha 1, beta 2 and the alpha 2, beta 1, but it's a weaker interaction. So the alpha it has 141 amino acid residues, and the beta has 146 amino acid residues. Okay, let's go ahead and take a look at an image of hemoglobin here. So we have our alpha 1, beta 1, we have our alpha 2, and our beta 2. So here you can see you have your alpha 1, beta 1, that interaction, that forms, so it's one subunit, 2, this is the third, this is the fourth. Now you can also make out in each subunit it has its own heme group. So here is the heme group for this one. And this one is a little hidden. You can see the heme group right here. Let me go ahead and, so it's right there. And you have the heme group right there for that one, and it's right there for this one, and here it is for that. Now I don't know if you can actually see the oxygens. It doesn't look like oxygen is bound, or maybe it is. It's hard to tell. But that's it. This is what it looks like. So alpha 1, beta 1, alpha 2, beta 2. That's it. Nice and straightforward, multi-subunit protein. You have this gap in between, which is going to become very, very important in just a little bit. Okay, so now our alpha 1, beta 1, and alpha 2, beta 2, okay, their respective interactions are quite strong. You have about 30 amino acids between alpha 1, beta 1, alpha 2, and beta 2 that are interacting with each other. So 30 amino acids interacting, that's a pretty strong interaction. Now as far as the alpha 1, beta 2 and the alpha 2, beta 1 down here, alpha 1, beta 2, alpha 2, beta 1, the interactions in this region and that region there, they are less strong. I'll put the interactions are less strong. Somewhere in the neighborhood of about 18 or 19 amino acid residues that actually end up interacting. When we use chemical agents to actually separate these subunits, as it turns out, they break up not into 4 separate subunits, or we can do that if we want to, but for mild treatment, what ends up happening is the alpha 1 and beta 1 actually stay together, and the alpha 2 and beta 2 stay together. So the interactions are quite strong between those subunits. All right, so hemoglobin, let's see. So hemoglobin, it has 2 major conformations. 2 major conformations. One is called the T state, and T stands for tense. This is where no oxygen has bound to it, so this is deoxyhemoglobin. So this is deoxyhemoglobin, and then there's something called an R state. In this case, R stands for relaxed. And this is the oxyhemoglobin. So when oxygen is actually bound to it, the more oxygen that's bound to it, the hemoglobin is in more of an R state. So when we say it has 2 major conformations, we're not saying it goes from T to R, boom. There is a transition. You have 4 oxygen molecules that are binding to the hemoglobin. So as one of them binds, there's some conformational change, and it starts to move away from the tense state, and it starts to make the transition over to the R state. So now it makes the second molecule of oxygen a little easier to bind. That second oxygen causes more conformational changes, shifts it more towards the relaxed state, which makes it more easy to bind the third oxygen. By the time the 4th oxygen molecule binds, it's completely in the relaxed state. So the relaxed state, the R state, has a high affinity for oxygen. The T state has a low affinity for oxygen. Now, these terms high and low, these are relative terms. We're not saying that this T-state hemoglobin has no oxygen attached to it. That's not it at all. In fact, it's about 60% saturated with oxygen. The difference is the R state is about 96% saturated with oxygen. So it isn't 0, 100. It isn't all or nothing. It's relative, degrees of saturation. The R state, very, very saturated with oxygen. That's the arterial blood. That's the red blood that has all that wonderful oxygen in it. The T state, about 60% saturation for the hemoglobin, that tends to be more of a bluish purple. That's the venous blood. That's the blood that's actually coming back from the tissues because it's delivered its oxygen. Now it's depleted oxygen. So the color actually changes. Okay, now the binding of O2, okay, so as we just said, the binding of O2 to a subunit of T-state hemoglobin causes a conformational change toward the R state. Toward the R state. And again, it is a transition. It isn't just boom, it goes to the R state. Toward the R state, which has a greater affinity for O2. So it begins to bind more O2. And the more O2 it binds, the more it wants to bind. That's this sort of domino effect, if you will, of this thing called cooperative binding, which we'll talk a little bit about a little bit later. Okay, so now